The discovery of antibiotics has revolutionized medicine by enabling effective treatment of many life-threatening bacterial infections. The fight against bacteria is again turning into one of the greatest challenges faced by our societies, especially with the spread of multi-drug resistant (MDR) bacteria. Acquisition of antibiotic resistance mechanisms is a natural process which is amplified by overuse and misuse of antibiotics, and inadequate or absent epidemiological surveillance in human and veterinary medicine, which has led to the dissemination of these MDR bacteria. Beta-lactams, due to their safety, reliable killing properties, and clinical efficacy, are among the most frequently prescribed antibiotics used to treat bacterial infections. However, their utility is being threatened by the worldwide proliferation of beta-lactamases, BL, with broad hydrolytic capabilities, especially in MDR gram-negative bacteria, GNB. Their dissemination is a matter of great clinical concern, given the importance of these pathogens as causes of nosocomial infections and of community-acquired infections, resulting in difficult-to-treat infections associated with high mortality rates and increased costs. The production of beta-lactamases is a commonly encountered mechanism of resistance in gram-negative bacteria, especially in enterobacterales. In healthcare settings, the activity of expanded-spectrum cephalosporin is challenged by extended-spectrum beta-lactamases, ESBLs, and plasmid-encoded cephalosporinases and carbapenemases, but ESBLs are by far the most prevalent mechanism in enterobacterales. The rapid and effective detection of antibiotic-resistant bacteria is a critical step for antibiotic stewardship and infection control. Despite technological improvements, the identification of pathogenic bacteria, as well as the detection of antibiotic resistance, remains complex and time-consuming, with time to results often in the range from 24 hours to 48 hours. Detection is mainly based on phenotypic approaches with antimicrobial susceptibility, using disc diffusion methods in the presence and absence of BL inhibitors. These conventional screenings are usually slow, requiring overnight culture, labor-intense, and require expertise for interpretation. The molecular methods, DNA microarray-based and nucleic acid amplification-based, either homemade or commercially available, remain the gold standard for the detection of CTX-Ms and other ESBLs. Molecular methods are usually slow, 1 to 4 hours, labor-intense, expensive, require specific equipment and trained staff in order to be implemented in any clinical laboratory. Lateral flow immunoassays, LFIAs, have proven useful as easy, rapid, and reliable confirmatory tests for detection of antibiotic resistance mechanisms, especially for beta-lactamases in gram-negatives. The NG Test CTXM Multi showed 100% sensitivity and specificity with isolates cultured on agar plates and was able to identify 98% of the ESBL producers identified in our clinical setting. These assays detect the presence of enzymes that confer resistance to expanded-spectrum cephalosporin. And even though CTXMs account for most of the resistance mechanisms leading to expanded-spectrum cephalosporin resistance, some rare enzymes may be missed. Biochemical confirmatory tests have been developed, which evaluate the enzyme's ability to hydrolyze beta-lactams. Biochemical tests mass spectrometry, UV spectrometry, electrochemical test. Even though these tests display good analytical performances, the biochemical tests are sometimes difficult to interpret. And MALDI-TOF, UV spectrometry, as well as the electrical chemical test require expertise, hands-on time, and specific equipment. Here we have developed an assay that takes advantage of the ease of LFIA and the universality of biochemical tests, and which detects the enzymatic activity of expanded-spectrum cephalosporin hydrolysis. This LFIA, called the Lfia Cft Test, is based on the structural discrimination of antibiotic and its hydrolytic product. we are going to present you the principle of this test, which detects cephalosporinase activity in a sample. The format of the test described in this presentation makes it possible to detect cephalosporinase activity from isolated colonies. The test requires a tube containing antibiotics and a sample cultured on a Petri dish. The antibiotics contained in the tube is first solubilized with an extraction buffer. The colony is then taken from the Petri dish using a loop and mixed in the extraction buffer containing the antibiotics. There are two cases: either the bacteria in the sample produces cephalosporinase, or the bacteria in the sample doesn't produce cephalosporinase. The tube containing the extraction buffer, antibiotics, and the bacteria is incubated for 30 minutes at room temperature. During this incubation, if a cephalosporinase is present, the cephalosporinase will degrade and hydrolyze the antibiotics in the tube. To detect cephalosporinase activity, an immunochromatographic test is used, which is based on the use of a strip. This strip consists of a first area on which the sample is placed, a second area consisting of a nitrocellulose membrane, and finally a last area consisting of an absorbent paper that allows the sample to migrate. An anti-antibiotic antibody labelled with colloidal gold is deposited and dried on the first zone. This monoclonal antibody has the particularity of only recognizing intact antibiotics. It does not recognize hydrolyzed antibiotics at all. On the nitrocellulose membrane, a first line is made by immobilizing intact antibiotics. This line is called the test line. A second line, known as the control line, is made by immobilizing an antibody directed against anti-antibiotics antibody. If the bacteria present in the sample does not produce cephalosporinase, the antibiotics present in the sample will be intact at the end of the incubation. When the sample is placed on the strip, these antibiotics will occupy all of the binding sites of the anti-antibiotic monoclonal antibody labelled with colloidal gold. During this migration, this antibody will therefore be unable to bind to the intact antibiotics immobilized at the test line. This antibody will be recognized and captured by the antibody present on the control line. In this case, only one line will appear. The test is negative. If the bacteria present in the sample produce cephalosporinase, the antibiotics present in the sample will be hydrolyzed at the end of the incubation period. Thus, when the sample is placed on the strip, these hydrolyzed antibiotics will not be recognized by the anti-antibiotics monoclonal antibody labelled with colloidal gold. During migration, this monoclonal antibody will therefore be able to recognize the intact antibiotics immobilized on the test line. Excess of this antibody will be captured at the control line. In this case, two lines will appear. The test is positive. The interpretation of this test is very simple and is done with the naked eye. The test on the left strip is positive, while the test on the right strip is negative. The Lfia Cft test is simple, fast, and inexpensive. The CFT test has been developed by the French Alternatives Energies and Atomic Energy Commission, CEA, and validated in collaboration with APHP and NG Biotech.